Okay, good. I was about to say good morning. It's not necessarily good morning for many of the people here. Hello and welcome to the third ENSO seminar. The third ENSO seminar also brings us to our third continent, as our speaker today will be speaking from Tokyo. Nathaniel Virgo is an assistant professor at the Tokyo Technical University and the Earth Life Sciences Institute there. After graduating as DPhil from the University of Sussex and also completing postdoctoral studies postdoctoral research at the Max Planck Institute for Biogeochemistry. Is that right, Nathaniel? In Jena in Germany, and then also completing postdoctoral research at the Ikegami Lab at the University of Tokyo. So this morning, Nathaniel, or this morning, again, sorry, my apologies for my temporal chauvinism or time zone chauvinism. Today, Nathaniel will be talking to us about enactivism and the origins of life, something that has played a sort of crucial role in the whole development of enactive thinking since its inception. In fact, for decades prior to the inception of the enactive approach more generally. So over to you, Nathaniel.
Hi. So I guess I'll just switch on screen sharing and get the presentation started. So here we go. Okay, so the talk is called Enactivism and the Origin of Life. I guess in the end, I'm not going to talk that much about the origin of life, but I will talk a fair bit about enactivism and kind of like my general approach to the central ideas in activism, because I think that in order to really address the origins of life, or rather not to just address the origins of life, but to really address the question of how a new autonomous agent can emerge where one didn't exist before, I think we actually have to push a little bit on the foundations of the field. So the central argument that I'm going to present is that well, enactivism, as hopefully most of us know, has its roots in a theory of biology, and I'm going to skip over differences between things like terms like autopoiesis and autonomy a little bit, because I kind of want to move away from strict definitions and get at the core ideas, which will be a theme of the talk. So the theory behind enactivism, whichever flavor of it you prefer, tends to be thought of in terms of strict definitions. This kind of goes back to Maturana and Varela, who have very strongly claimed that systems are either autopoietic or they're not autopoietic, and there's no, and autopoiesis means life, so there's no shades of grey when it comes to life. But this, I think, makes it a little bit difficult to think about the emergence of autonomy. So most enactive approaches to the origin of life they're really concerned with the minimal instantiation of autopoiesis. You have to start with a whole in order to then progress to anywhere else. But if you're interested in the origin of autopoiesis, then it's a little bit tricky, right? Because you need to have something that's somehow nearly autopoietic, but not quite. Otherwise, it's hard to think about how you can get there. So the proposal that I'm going to make in this talk is that we kind of should actually be trying to soften the foundations of enactivism. We should be moving away from these kind of very strict definitions, and we should be grounding it instead in observations about the physical world. So it's not that we should work without definitions. It's not that we shouldn't be clear what we're talking about, but that it's always important to do that. But we should work towards a formal definition as the end point of a process rather than as the beginning. And I'm going to try and argue that that looking at enactivism this way actually leads you to see the world differently. We move away from seeing life as being a collection of distinct agents to being this like kind of interconnected network of processes that takes up the whole planet, and then within that many different agents emerge and have different degrees of autonomy from one another. So that's what I'm going to try and claim. So my motivation for doing this, as I say, is the origins of life, and it's worth saying just a little bit about what that means for me. So the question in Origin of Life is that we know that about 4 billion years ago, a little bit more than 4 billion years ago, I guess the world was all molten lava, and about 3.8 billion years ago, we know there was life on it. So, so at some point, you go from this transition from a lifeless but chemically active world into a world with a biosphere. So, you know, you can look at this as a timeline. You start with no life and you end up with modern cells. And people pretty much agree that at some point just before modern cells was this thing called the RNA world, which is quite a lot like life today, except that RNA plays a far bigger role and peptides play a lesser role. But RNA world is pretty far from nothing, because RNA is a very complicated molecule. It's very difficult to make. So there's this whole extremely mysterious gap in between no life and the first, what we think the very earliest thing that we can look back at is. And so all these things, metabolism, large molecules, membranes, and everything had to happen at some point, and the point is we just don't know how. And so my work is about trying to understand whether there are general principles that would take you from no life at all to something here, which is whatever it is that has the ability to become more complex over time and eventually become a cell. So a lot of work in Origins of Life is concerned with how to get the right molecules. How do you get the amino acids? How do you get the nucleotides? How do you polymerize them into the right macromolecules? And then, you know, the hope is that once you've got all the right molecules, there'll be some way to put them together into an organism. But enactivism, for me, the central insight of enactivism, the thing that I take from enactivism and want to put into the Origin of Life as a field, is that life isn't made of molecules; it's made of processes. And the rest of the talk's going to be kind of about understanding what that means. So Marek gave a talk two months ago, the first ENSO seminar, and in one of the things he did in that was he made a distinction between the structure that comes from the combination of many elements versus structure that emerges by the differentiation of something that was there before. And so what I want to say about the origin of autopoiesis is that we shouldn't think of it as being a combination of elements. We should think of it as happening through the formation of a distinction from a homogeneous initial state to one in which there is an agent distinct from its world. So yeah, how do we try to achieve a really inactive approach to the not just to the origin of life, but to the emergence of autopoiesis? So it's hard not to start with the definition of autopoiesis if we want to get to this. This is one of the key components of enactivism, or one of the key works that led to enactivism. It's this book, Autopoiesis and Cognition, by Maturana and Varela. And they wrote this definition, which kind of sounds like a good, solid, formal definition. It says an autopoietic machine is a network of processes which, through their interactions, continuously realize and regenerate the same network of processes, and at the same time constitute it as a concrete thing in space. I skipped over some of the words, but that's what this definition sort of says. And it's not really a formal definition. We shouldn't look at this as being being a really strict formal definition. I think we should, and it was originally quite tentative, so we should think of this as being an insight. Like, this is a thing that life is, and it is important that life is this type of thing. So, and I see it kind of as a promise of a theory that will later be worked out. I think for me that's the best way to think of it. And so a lot of the work that I did back in my PhD and that I'm sort of starting to come back to now is about trying to understand how to pan that out in a way that makes sense, that helps you think about the origin of life. So for me, processes, well, this definition is in terms of a network of processes. And to me, processes include both things at the basic chemical level. Metabolic reactions, like a step in the Krebs cycle, is an example of a process. Transcription of RNA, just another chemical reaction, much more complicated, also a process. Transport of ions across a membrane, so processes, you can have transport processes. It's quite clear that can be called a process. But I think you should also include things like developmental processes, like growing a tooth, and behavioral processes like learning or thinking. The key insight for me is that these processes are all part of the same network. The cell is thought of as a network of processes, and they're not just at the low level, and they're not just at the high level. They're kind of all the way in between. So life and mind are tightly interlinked, at least, and that's one of the key insights of enactivism. So autopoiesis says that a cell is not just a thing that happens to be a thing that has a membrane and it has DNA in it and it has all these molecules in it, and those things engage in processes. Autopoiesis says that the organism is the network of processes, and if we want to understand the origin of life, we have to understand how you can get a network of processes that is this tightly coupled, that kind of all really interlink to one another, all reinforce each other, all end up with this spatially localized thing. So that's my kind of brief summary of autopoiesis. But now what I like to do is I like to look at other things that exist in the world that seem to have similar properties, that aren't living cells, but they share some of these nice properties that this definition points at. I'll say a little bit in a while about why you would want to do such a strange thing. But first, let's have a look at this hurricane, diagram of a hurricane. We can see here that there are lots of processes that happen in a hurricane. There's the evaporation of water from the warm sea surface. There's this kind of cycling of winds that's caused by the Coriolis effect. There's the upward convection of air inside the eye. There's the outward motion of air. There's the radiation of heat into outer space at the top, which is what keeps it going, because the whole thing is a heat engine. It transports heat from the ground to the top of the atmosphere, and by doing so, it extracts usable energy, and it uses that energy to construct itself. And you know, if you say this idea to somebody who's an expert in hurricanes, they say, yes, of course, that's what it is. It's exactly like a living cell. They get very excited. So I think that's really interesting. I think that's really useful and tells us things, because it's an example of something that has the same property of a cell, but it's much simpler than a cell. It's much easier to understand. You can look at all these processes and see how they really do interlink. But there tends to be there is this tendency for people to sort of push back against it and say, no, that's not what I am interested in. I'm interested in life, and I want to have a nice hard boundary between life and non-life, and that's fine. I mean, for many of the purposes of enactivism, that's a useful thing to do. But for me, looking for the emergence of life, I'm really interested in the blurry part of the boundary between life and non-life, because if we can see where that distinction becomes blurred, then we've really got somewhere. So that's why I'm interested in thinking this way, and I hope I can convince you that it is useful. So yeah, it's very interesting to think about what things like typhoons share with living cells. And for me, it's more interesting to think about that than to think about what the differences are, although, of course, the differences are also enlightening. And so this example of a hurricane is chosen carefully. It's not just a randomly picked example, and it's not a candle flame, it's not a wave on the sea, it's a hurricane. And I chose a hurricane because it has a couple of properties that are relatively rare in the non-living world. So one of these properties is precariousness. And this is a notion that's been very central to enactivism. Precariousness essentially means the possibility of death. And a hurricane, if you were to perturb it too much, it would lose its shape. It would no longer be able to continue existing. It would no longer be able to continue extracting work from that temperature difference between the sea surface and the upper atmosphere, and it would go away. It would disappear. It would disintegrate. And the second property that it has is what I call individuation, and there are other people who use the same word to mean the different things, so one has to be careful. But by that word, I mean the hurricane has the property that it not only exists as a distinct thing in space that you can point at, but it's responsible for doing that by itself. The fact that there is one hurricane in one place and another hurricane in another place is because of the dynamical interactions that happen between the processes that constitute those two distinct hurricanes, and not because of some external individuating force. And I'm going to go through this fairly quickly, but it's useful to see some other examples of things that have those properties to different extents, or maybe don't have them at all. So these things are in the class of what we call dissipative structures, which is a sort of fairly loosely defined thing, but it means a structure that you find in physics that persists because it extracts usable energy from its environment. So you get chemical traveling wave patterns. You can have fire. You can have sand dunes, and so on. You can have human beings are also in the class of dissipative structures. And they're to be distinguished from things that are not dissipative, that are just sort of static in some way. I actually have, since I made these slides many years ago, I've become much less convinced that there's a real distinction between these things, but that's a discussion for the physicists. But roughly speaking, you can divide the world into dissipative structures and things that are not dissipative structures. The dissipative structures are the ones that exist by extracting energy from their environment. And some of them are precarious and some are not. So of the dissipative structures, some of them will come back if you try to make them go away, and some of them won't. So a sort of standing wave in a river, it doesn't matter how much you perturb it, it'll come back again. But a hurricane, you can perturb it. A flame, you can blow it out. And so my rough definition of precariousness is that the conditions required to form the phenomenon are different from the conditions required to maintain it. So in the case of a hurricane, it only forms if you get quite a specific thing. You have to have a very low wind shear. You have to have this large collection of thunderstorms all happening at the same space. The wind has to be sort of swirling a bit. And once you've got those kind of conditions, you get this sort of proto-hurricane that spins up and becomes a fully developed hurricane. But that fully developed hurricane can then move around and persist in areas of the sea where a hurricane would never form. And similarly, a human being can be formed only in the very specific circumstances of having a fertilized human egg in a human womb, and then a family to feed it as it grows up. But once that's happened, it can then go off, a human being can then go off and survive on its own in the forest if it needs to, or it can get a job and live in a city or whatever. There are many other places you can survive than places you can be formed. And so that's something that can be true to degrees. And it's a pretty common property. So most of these examples that I gave have it. And in fact, it's actually sort of hard to find ones that don't, and they're sort of debatable. I used a lenticular cloud here, which is a type of cloud that, well, I'm not going to go into the details of that. Debatable cases. So yeah, it comes in degrees. So the other thing was individuation. So individuation, some types of dissipative structure are formed into distinct individuals that are the kind of these spatially localized things in space, and it's these things, the ones that have both of these properties, that have something similar to autopoiesis. I'm going to skip over some of the details. Individuation is something that comes in degrees as well, and this is also true in the living world. So this is something that needs to be dwelled on a little bit, actually, which is that the question of why there are individual organisms is not an obvious question, right? Because biology is so clearly divided into organisms that we don't often stop to think why it should be. But if you do think about it, it's kind of remarkable. And the fact that the exceptions are rare, like this, the humongous fungus is the world's largest living organism. It's a mushroom. It's a fungus that has a network of whatever you call them, fibers underneath the soil, and it takes up a large geographical area. So people call it the largest organism, but is it really an organism? Because if you cut it in half, it probably wouldn't care. So it kind of lacks this property of individuation somehow. And there are other things in the living world that in various ways lack the property of individuation. But so it's not obvious that that's a question, but I think it is a question. Why is it that that happens? Why is it important? So yeah, an individuation doesn't imply a hard separation between an individual and its environment. So this is actually kind of important. So the hurricane example, one of the objections that people often give to it is that, well, it doesn't have a membrane. And the fact that the living cell does have a membrane is really important for the way a living cell works. But in terms of this kind of network of processes idea, the idea of autopoiesis, I don't think the membrane itself is actually all that important. it's just one of the generation of the membrane is just one of these processes, and it contributes to maintaining the other processes by in many ways by containing things in a spatially localized area. But it's not the only way you can remain spatially localized. Hurricanes do it without membranes, and I find that interesting and worthy of note. So yeah, so you have some dissipative structures, including living organisms, that have both of these properties, and these are the ones that all have analogues of metabolism. They all have some kind of anatomy, because once you're spatially localized, you're not homogeneous anymore, and they all have behavior, and that's quite interesting to talk about that. But I don't have time to go into it in depth. And you can divide this diagram down further and try and position life somewhere here, not quite always individuated, has genetic material that produces membranes and so on. Cells obviously are in there as well. So yeah, the summary so far, just to recap where I've got to. Actually, this is I'm going to have to skip over a lot of the talk, I think. But the summary so far is that the enactive approach to biology usually starts from this kind of abstract definition and argues in kind of logical terms from the consequences. But that made it difficult to think about the origin of the first kind of autonomous agents, or the first autopoietic systems. So I'm proposing a more observation-based approach where we kind of catalogue what we see in the physical world, and we figure out where biology fits into that. And if we, having kind of taken this rather different perspective for a little while, if we go back to the traditional enactive approach, we possibly get some new insights. So a key notion in Origin of Life is operational closure, and that's the idea that associated with an autopoietic system, or in some way an autopoietic system, is a set of processes that is self-maintaining. And the way that you sort of start to cash out that idea of being self-maintaining is that every process enables or enhances at least one other process in the system, and also is enabled by at least one other process in the system. So it all kind of links together into this network, which we call operationally closed. And you should note that this diagram is just a sketch to help the intuition. I've actually tried to produce the corresponding diagram for real systems, and it's really hard, and you sort of realize it doesn't really work just writing enables as being arrows, and you have to make a more complicated diagram, and it's really not obvious how to make it all work. Let's see. So, there's some really interesting stuff about extended autopoiesis that I published with Matthew Egbert and Tom Froese a long time ago, but I'm going to skip over it in the interests of time. The point here, though, is that this network of processes isn't necessarily confined to what's inside the organism. It also includes feedback loops through the environment. So this is kind of the idea here. There is that you can kind of look at the extended mind ideas, and you can extend those to autopoiesis and say that this network of processes that maintains an organism actually includes things that happen in the environment, like in the case of this spider, which injects its digestive juices into the cocoon and then later comes back after the digestion has happened outside the spider's body and eats the pulp that's produced. And the point is that that's something that's enabled by the spider, but it also enables all the things to happen inside the spider. So I want to get to the modeling part of the talk, and I'll try and get through this in sort of 5 minutes, because this kind of gets me to my point about the origin of life that I hope to be able to make by the end. So this is an example. I didn't invent it. This is an example of a dissipative structure. It's a simple simulation of self-organization in a chemical system. I hope the bandwidth is good enough that you can all see this this animation. So basically, what you should think of this as being a set of chemical reactions taking place on a surface. There's red and there's blue, which I've called U and V here. There's green and there's blue. Sorry, I should say. And the blue stuff is fed in to every cell throughout the time, and the green stuff is a chemical reaction that is autocatalytic, meaning that this reaction happens to create more. The reaction it requires green stuff to be there, but it produces more green stuff as the reaction proceeds. So it kind of grows exponentially, or actually super exponentially. And at the same time, these chemicals diffuse across the surface. So I haven't explained those details very thoroughly, but they're not really that important. The point is that this is just an example of a dissipative structure, and it happens to have the properties of being individuated and being precarious. So it makes a good minimal model of the thing that I'm trying to get at. And depending on the parameters, you can form all sorts of different patterns, and sometimes they're much more stable than others, and sometimes they're not individuated, and sometimes they are. And well, you can go through this thing, and you can look at the processes and how they fit together and how they work, and you can start to try and draw this diagram, but it's only ever a cartoon sketch, because really drawing this diagram is really hard, because it's hard to identify whether the same thing happening in different parts of space is the same process or two different processes, and this kinds of things are hard to resolve. So the interesting thing is that you can then extend the system by adding other processes. So here we have the same system, except that I've added another chemical reaction, where now if I change the colors, unfortunately, so the stuff that was green before is now pink, but now we have some green stuff that eats the pink stuff. So in direct terms, being eaten is bad, but you can see that what's happening is that the pink spots are forming a tail of green stuff. So the tail is eating the pink stuff. And in eating it, it causes it to move. And it turns out that moving is actually good, because moving means that you can find more food. And so I want to show you a very quick demonstration. I think, yeah, we started 5 minutes late, so I'm only 2 minutes over my time. So this is exactly the same system, again with slightly different colors if it runs. There it goes, yeah. So this is exactly the same system, but with different colors. And what's going to happen, so this G at the top that was equal to 1.8 just now, that's a parameter that just says how easy it is to survive in the system. That parameter is how much food gets fed in. And you can see that when things were good, when G was high, the blue spots did pretty well. They were colonizing the system, and the red was a kind of annoying parasite that was eating them. But now as G starts to drop, what happens is that the blue spots, because they don't move, they become unstable. They're sitting, they deplete the food locally where they are, and they become unstable and they disappear. So it's precariousness in action. But the ones with the tails are always constantly moving into new areas of food, and so they survive. And you'll see in just a few moments that all of the blue spots will die, or disintegrate, whatever you want to call it, and only the ones with tails will be left to survive. And so it's a bit of a parable, I suppose, but it's kind of interesting from an origins of life perspective, because now the system is in a stage where the only way to survive is to have both blue and red. So it's kind of pulled the ladder up from under itself. It started out simple, and now it's more complicated. But the world has changed, so it can't. You can't go back to being simple again. So if you were to observe this without having seen the initial state, you would wonder how these things got there. And so this is an example. I sort of said earlier that we should think of the network of processes in autopoiesis as including processes on the chemical level and processes on the behavioral level. And this is a really good example of that, because here we have these things that can only survive because they move. And movement is a kind of very basic behavioral level process. And so essentially you have this network of processes that I've sketched here, where you have two chemical processes and one behavior level process, and the network contains all of them. It crosses levels. So I think that's pretty neat. And so finally, I've got a few slides on the concept of open-endedness. So this is kind of like where I want this sort of research to go in the future. So the question is how. So a lot of work on origins of life from the enactive process from the enactive perspective, focuses on the understanding the minimal instantiation of autopoiesis. People like to think about the minimal one perhaps because that seems like the easiest one to get. But the question is, how do you go from simpler to more complex? And I think that's a pretty interesting question. And I think the resolution to it is, roughly speaking, this: that in a small network of processes, like the ones in the model that I just showed you, where there were diffusion and there were a few reactions, but those are the only processes that could happen. In a small network, there's not really much capacity for change. There's not much that can happen. But in the real physical world, and particularly when chemistry comes into play, there's an infinite number of processes that could occur if only conditions were met. Now, if there was some salt, some vinegar and some baking powder in this room, then the process of making CO2 could occur, but there isn't, so it doesn't. And that means that you can have kind of cascade effects, where if some new thing enters the system, some new process starts to occur, that can suddenly enable processes that weren't enabled before. And if there happen to be these loops in the network, then that's when you can get the emergence of a new organizationally closed system. Or you can have the extension of an old one. So this would be a kind of permanent change. You can permanently add something to an autopoietic system if it's in this kind of open-ended situation where there are many possible processes that just haven't started happening yet. So I think this is basically how complexity can increase. If you're in a complex system to start with, there are always new processes that can start happening that weren't happening before. And so ultimately, I think this is how you can get to evolution. So at the moment, I'm very excited by understanding how you can go from this kind of piecemeal addition of new feedback loops to actual DNA-style evolution. And so to be able to understand this complexity, we have to move away from minimal models. So that was kind of that's my point towards understanding the origin of life. So this is my last slide. So to summarize, I've argued for taking the key insights of Maturana and Varela, but giving them a less abstract grounding, one that's more grounded in empirical observation of the world, kind of cataloging things and observing things and seeing if we can work towards a definition that really captures what's going on. And this approach kind of sees processes as dynamic. Processes are things that can start and stop and change over time, and that's basically how you can get the emergence of autonomy. And then finally, when you start thinking about these things in the softer way, you start to see a world that consists of a single network, and there are many interconnected agents within this network. And I sort of forgot to mention this in the slide, but this connects really nicely to Matthew's talk last month, Matthew Egbert's talk, because he talked about habits as being a kind of a habit as being a sort of self-maintaining thing in its own right, and that's a fairly that's a current notion in enactivism. But there are also cases where behavior or habits can be vital for the survival of the organism, and so I've always felt there's a kind of tension between those two sorts of notions. When is a habit an autonomous agent in its own right, and when is it part of the autonomy of the organism? But if you move away from these hard definitions, this is an agent and this is a different agent, and you start to see it as one network where things have a kind of degree of autonomy from one another, then it starts to make sense, and you can kind of see that there are that you just don't need to make that distinction. It can be more or less of one or the other. So yeah, that was a slightly tangential place to conclude, I suppose, but that's the end of the talk. So I hope got something useful out of it.
That's great, Nathaniel. Thank you very much. I really enjoyed that. So I'll just I'll allow us to move back and forth a little bit now, because I'll give the viewers just an opportunity to type. So if you've not been in an ENSO seminar before, hopefully you will have, or even if you have, if you haven't been at an ENSO seminar before, then you may not know that there is a means to ask a question of Nathaniel through the Q&A app. So if you sort of, I think over towards the left-hand side of your screen, there's a Q&A icon, and that will allow you to type questions, which hopefully Nathaniel will be able to see. And we'll be able to respond to if there are any immediately. If there aren't many immediate questions, the discussion can still continue on the ENSO Seminars webpage, ensoseminars.com, where if you click through to the specific page for Nathaniel's presentation, there'll be an opportunity there to add comments or questions, which Nathaniel and others will be able to respond to as well. But thanks a million for that, Nathaniel. That was really clear, and I enjoyed it. Now I'm asking questions from a somewhat naive perspective on this to an extent, but I have a couple of questions that I'd really actually like to hear you respond to, particularly on that last point about individuation that you mentioned, because once you begin to dilute the importance of individuation, things get very strange very quickly. And on the one hand, I'm fairly convinced by your argument that we shouldn't be too rigid in our requirements of, hard individuation, that sort of very clear and distinct boundaries between one organism and another. But on the other hand, I'm then somewhat at a loss to properly understand what the importance or role of individuation is in the definition or pointing to of life. So if it basically, does individuation really matter? And if it does, how does it matter? Can we say, I know you can't, definitive answers might be a bit of a stretch, but if you could say a little bit more than that, I'd be very grateful.
Sure. Okay, so I guess individuation means multiple different things at the same time, and it's sort of interesting that those things come together with each other. So on the one hand, from the kind of origin of life point of view, there's an extremely important practical consequence of individuation, and that is that when one organism dies, only that organism dies, and another one doesn't die. So if you have heritable variation between organisms, that's what you need to get evolution, basically. Like if a new process would happen here and it would affect everyone, then it could never be a heritable trait. So if you want to get evolution, you need individuation. That's one thing. But another reason it's important is the kind of distinction between the individual and world. I guess I don't have the answers to these things. So I guess, one is just to note that it happens, right? To note that there is individuation in biology. It's everywhere, right? It's at the level of cells. It's at the level of multicellular organisms. It's at the level of colonies. Sometimes it breaks down. Sometimes you get weird things where you're not sure if it's one individual or two. But it's remarkable that it is so common. And it's remarkable that it also happens in the physical world. So it's a kind of observational question. There is this thing in the world, and can we understand it? Why is it like that? But I think also, ultimately in enactivism, you want to get to phenomenology, right? You want to be able to say there is an agent and there is something that it is like to be that agent. And I would love to be able to eventually see. So I guess a lot of people think that autopoiesis is the thing that gives you phenomenology, right? Or autopoiesis plus something additional like adaptivity maybe is what gives you phenomenology. And so then it's a question that I don't have the answer to. Is individuation an important aspect of that or not? So.
Okay, cool, thanks. I think that I particularly like the idea that, or what was particularly convincing, I suppose, there was the notion that given that you've made the argument that complexity can help make a system more stable, individuation is one of the ways of making a system more complicated too. It's sort of, once you draw boundaries, you actually get an opportunity for things to interact rather than just diffuse, I suppose.
When you come to forming a membrane, that's an enormously important way in which the cell is able to be complicated, and a lot of work in origins of life both in the enactive origin of life and in just origin of life generally, focuses on how to achieve that, because if you didn't have the membrane, things would just float away into the environment and they wouldn't interact, and they would interact with water, and that would make them go away. Yeah, it's an important question.
Very good. And I again, sort of then one just sort of final question. I think this one might be fairly brief, and it's just to do with when you discuss complexity and the fact that when basically moving away from minimal models, that the introduction of chemistry really complicates things and enables a sort of richer range of possibilities. Do you see any difference between the kinds of complexity that something like chemistry enables and the kind of complexity that something like behavior enables? Or is the richness of a behavioral culture, for example, performing almost an identical or certainly very analogous thing that that a chemical culture will be doing?
That's an amazing thought. I really like that idea, and I hadn't thought of it before. So yeah, I mean, because normally, normally the way that I think is in terms of chemistry, it gives you, what chemistry gives you is this kind of ability to be, or particularly in organic chemistry, molecules are kind of, they're in this local equilibrium type of state. So that the molecules are, they can be put into a state and remain in that state for really long periods of time unless another thing comes along and interacts with them and changes them into a different state. So when you're in the business of comparing chemistry to physics, that's kind of like one of the important aspects of chemistry. And the fact that chemistry has that property means that you can have one set of processes going over here and a totally different set of processes going over here, because you've got a different set of catalysts. So then the question is, well, so on the other hand, when you get up to having brains and learning and different behavior, is that doing the same thing? And in a way, yeah, I think it is, because if I've learned to do a different bunch of things, then I guess like a molecule, I've been put into a state and I'm staying in that state somehow. I mean, it's kind of playing a similar role for totally different reasons, I guess. I mean, I'm just completely just thinking on my feet. But yeah, I like the question.
Very good. Okay, excellent. Thank you very much. So I think maybe we should leave the broadcast there and allow the discussion to move to the website and sort of unfold over the kinds of timescales that website discussions tend to, rather than trying to cram everything into this brief period of time online live. So it just remains for me to thank you again, Nathaniel. That was a really good talk. Really, I'm grateful for you for giving the talk, and to invite everyone to join the discussion at ENSO seminars. And we look forward to the next seminar, which will be early next month. And now I should know without checking, but I'm just going to double check that our next seminar will be given by Aaron Agman from Indiana University on the ontogeny and adaptivity in a model protocell. So following the same development of life theme within enactivism. So thanks very much, Nathaniel, and everyone else, we shall hopefully see you next month.